Hello, my name is Evan Laine, and welcome to the launch of SpecterCast. This is our first show. SpecterCast is a podcast offered and presented by the Arlen Specter Center at Thomas Jefferson University. The mission of the Arlen Specter Center is to facilitate and promote public service and civic education in a cross-disciplinary, nonpartisan setting. The work of the center includes programming inspired by Senator Specter's long-term interests and long-term career accomplishments. SpecterCast's specific mission is to share with the university and the public the exciting research being done by our professors, scientists, and administrators in the areas of medicine, social sciences, architecture, design, and economics. Today's podcast is called Cornering COVID-19. Obviously, we'll be dealing with the ongoing pandemic that we're all, using a charitable word, experiencing right now. Today, I'll be joined by my co-host, Professor Raju, who will introduce himself shortly. But our guest today, our honored guest today, is Dr. Mark Tykocinski. Dr. Tykocinski is Provost and Executive Vice President for Academic Affairs at Thomas Jefferson University. He's the Dean of the Sidney Kimmel Medical School, which is associated with Jefferson. He's also served 5 years as President of Jefferson University Physicians. He's a scientist. He has contributed in many fields, including molecular and cellular immunology, cell surface engineering, biologics, and other innovations, period. What makes Dr. Tykocinski especially valuable today is his knowledge of the research ongoing by Jefferson scientists and professors on the treatment and cure of coronavirus. In addition, he is, in his own laboratory, working on some very innovative new treatments to minimize the effects of the disease. At this point, I'd like to introduce my co-host, Raju Parakkal, before we begin questioning Dr. Tykocinski.
Thank you, Professor Laine. I am Raju Parakkal, a professor of international relations at Thomas Jefferson University, and we are quite happy to have Dr. Tykocinski join us for this inaugural podcast of SpecterCast, a brand new series of podcasts that we are launching with this particular episode, as Professor Laine just mentioned.
Thank you, Professor Parakkal. And now, Dr. Tykocinski, thank you so much for being here today to help us in this very confusing and very distressing time. I'd like to take a two-step approach in our questioning. First, I'd like to talk about vaccines, which of course is prevention, and then move on to treatment, which you're particularly involved in your own research. In regards to vaccines, I'd like for you just to basically give us an idea on how these vaccines work.
Take a protein from the surface of that microbe, and that itself becomes the vaccine. And hepatitis B is there. And then there's this recent concept of taking nucleic acids, DNA or RNA, these genetic elements that code for the viral protein, the key viral proteins, you introduce those DNA or RNA fragments. And to date, there have been none of those that have actually made it into the clinical marketplace, if you will.
I've heard people who get excited about vaccines. They say, I'm going to, and this even flew from some of my students, I'm not going to put the virus in me because you're going to make me sick with that vaccine. I've heard, what, does that make any sense to you?
Well, the fact is, once these vaccines have gone through their initial trials and have been validated, they are just remarkably safe. The adverse effects you might get for one that's already made it into the marketplace might be a little fever, muscle ache, and that's usually not the case either, or some pain at the injection site. Occasionally, there might be an allergic reaction to some other component in the vaccine, but these vaccines that have been validated are safe, and it really is an incredible disservice out there, in the webs, in terms of putting those vaccines, out of place. Understand that vaccines do have to be validated. So every candidate vaccine still has to go through that clinical trial, and sometimes you will get paradoxical effects, and then that doesn't make it out into the clinical arena.
Let's talk about Jefferson then. I've been reading a little bit about Coravax and some of the promotional material coming out of Jefferson. Tell us what it is and what's going on there.
Sure. Let me put it into a little context. COVID-19. There's been a whirlwind these past few months. Really, an avalanche of candidate vaccines and therapeutics. And that's along with an avalanche of claims, perspectives, actions. Some refer to it as a COVID-19 infodemic. And it's really up to us and, frankly, webcasts like this one to try to start to tease apart all the hype that's around it. And, some of the hype is actually well-meaning in many cases, but it is driven in part sometimes by opportunism and happenstance positioning. Company, pharma or biotech happens to have a drug related to some, agent, and now they say, well, that could be repurposed for COVID-19, and suddenly that sort of gets all the attention. So I think an example of that, and this is the counterpoint to what we're doing here, literally over the last 2 or 3 weeks, you've heard a lot about the DNA and RNA vaccines. And that's attracted a ton of attention, a lot of funding, including federal dollars that are now going to be pumped into that. But we've got to be aware that to date, to my knowledge, there hasn't been an RNA or DNA vaccine that's actually made it into the clinical marketplace. So while they're clearly incredibly elegant in concept, right? If you just had to take sort of a DNA or RNA stretch and put it into the person, it goes into the person and the person's own cells make the protein that drive the immune response. That's true. And it's also true of another set of vaccines that are out there, the adenovirus vector-based vaccines. You've heard a lot about in terms of University of Oxford and AstraZeneca, J&J, et cetera. They've really been in the limelight, but here too, there's, to date, to my knowledge, there's been no adenoviral vector vaccine that's demonstrated that it can prevent disease in humans. So that puts the work of Matthias Schnell and the Jefferson Vaccine Center into context here. So we've called it a Coravax, C-O-R-A-V-A-X, for corona rabies virus. That sounds a little strange, but it's built on the platform of a rabies virus vaccine that's been proven effective and safe in hundreds of millions of people around the globe today. So this is developed in the Jefferson Vaccine Center under the leadership of Matthias Schnell. Dr. Schnell is also the chair of the Department of Microbiology and Immunology here at Jefferson. And what he's done is something elegantly simple. He simply said, if the rabies virus vaccine works so well and is safe, let's just piggyback onto it and express on its surface an extra protein. And that's the spike protein of the SARS-CoV-2, which is, the COVID-19 virus. It's that alone makes it, in my mind, a strong shot to, be a winner here. And in fact, back with 2 other coronaviruses, with SARS and MERS, and also with Ebola, the Schnell lab had validated this and had gone as far as putting it into non-human primates. Now, those epidemics subsided, and it never went into the human context.
So I just understand it, Doctor, for the lay audience,
sure.
Which is basically me. You're taking an existing rabies virus and you're altering it to make it into a COVID-19 or to look like a COVID-19?
You're altering it to add to its surface one extra protein. That one extra protein is a protein from COVID-19, the virus of COVID-19. But it's not just any protein, it's a key protein that is expected to drive a protective immune response against the native COVID-19 virus, called SARS-CoV-2. The other thing that has us, enthusiastic about the possibilities for this virus is that it's scalable. It's proven scalable because the rabies virus vaccine, the attenuated rabies virus, has been shown to be there. But importantly, it's amenable to storage. You can keep it at room temperature. It can be stored for months and transported. So this is ideal for the developing world. And we just came out with an announcement last week that one of the largest vaccine manufacturers in the world, called Bharat in India, which is, I believe, the leading manufacturer of rabies virus vaccines, has now licensed this for the developing world. And they're in fact converting a good deal of their production instantly, even before it's been validated in human trials, to starting to produce and scale up this CoRvax vaccine. So
that's something.
That's exciting.
Yeah, Dr. Tykocinski, that's something that we are interested in learning more about in terms of Jefferson's international collaborations and outreach, and why you also think that this is so particularly important for when it comes to cost structures and different socioeconomic conditions.
Yeah, so I think you're touching on sort of a very key question here, is scalability. Can something be made, and if it's scalable, can it be at a price point that is meaningful for the vast segment of the world that is not, that isn't going to pay the kind and doesn't have the kind of dollars to pay for those vaccines. And then the other point I had made was can it be distributed? I mean, you're now getting it to places where you don't necessarily have refrigeration and the kind of transport systems that allow that to go there. So a number of the vaccines, that really have been part of the, have triggered the hype and the excitement, they sound wonderful, but the question has to be asked, even if they are validated, and I certainly hope they will be, because we're going to need more than one type of vaccine here, to be meaningful across the globe. What is its cost structure? And is this something we're going to be able to deliver worldwide in the way that we have to do that? Now, mind you, in the case of Bharat, they've just licensed it for the developing world, and we're still looking for a partner in terms of getting this commercialized and developed for the developed world.
Doctor, when you say validated, you've used the term a number of times. What exactly are you talking about?
Yeah, so there are two parts really. Validated means, one, that it is effective. Will it stimulate a protective immune response? And the main thing we look for is the development of antibodies, neutralizing antibodies that will neutralize the virus. And the second piece is safety, making sure that there have been occasions in the past in the development cycle of a given candidate vaccine that it stimulates the wrong kind of response. And so we need both efficacy and safety data for that validation.
We've heard a lot about timing, and different promises have been made. We're not going to get into who's making them, but different promises have been made. What does timing have to do with all this? Is this something we do tomorrow, next week, next year?
Well, I think we've been guided by sort of experience in the past, and experience in the past generally has given us longer timelines. So you'll hear people saying that optimistically it'll be 12 to 18 months before we have a vaccine. The situation now is a little different because I don't know that there's a precedent for the kind of worldwide engagement. We've got, I don't know the last number, but it's probably over 80, 90, maybe even over 100 vaccine candidates that are being worked on around the world. And, and some of them have already, have already gone into the first humans, and there's some inkling that might be there. So I think the timeline could be shorter here. I mean, it's really hard to say. And I think it's going to vary considerably by the approach. One of the things, that we like about Jefferson's candidate amidst that, a family of possibilities, is that it's built on something that already has been out there. So we sort of know how to work with it. We're working with a company that has vast experience in that. And so we're hopeful that, that it'll be faster than that 12 to 18 months, but it's really hard to say at this point.
I'd like to, unless Raju has any questions on vaccine, to pivot into treatment. Dr. Parakkal, anything on?
Yeah, I just wanted to ask Dr. Tykocinski, to the extent that it's possible for him to explain, how does the Jefferson solution work in relation to other comparable solutions out there?
Yeah, so I sort of listed the different vaccine approaches. The Jefferson solution really fits into the class of vaccines that are like polio, for example, right? Where you had the Sabin and Salk vaccines. One was attenuated, one was a killed vaccine in this instance, and this is sort of tried and true. The other ones that I mentioned, some of the others that I mentioned, including the adenoviral vector-based ones and the RNA DNA-based ones, again, these are very elegant solutions, and I'm really hopeful that they'll work. But again, these haven't made it into the marketplace yet. So there's a bit of rolling the dice there, and that validation will require both the efficacy side and the safety side.
And I assume that the price points for them would also be higher than what Jefferson plans to offer?
My guess is, now again, there's large amounts of dollars, federal dollars coming from our country and others, which will, so I haven't, can't tell you where it's going to leave the final price point, but in generally, if it's issues of cost to manufacture, we're building on a rabies virus vaccine that has been distributed across the developing world. So, and that's, so it has been, priced to be, just for that purpose. So the answer is likely yes.
Likely.
I think something based on a virus and attenuating or inactivating a virus is, I would guess in general terms, would be, easier, lower cost manufacture.
Doctor, before we move on to treatment, just one last question on vaccines. How deep are we, Jefferson, into the verification process?
Sure. So Dr. Schnell and his team have demonstrated that in animal models that it will develop a antibody response, which is a good step. We're probably just a couple of weeks away working with one of the best animal models that is out there now for COVID-19. And we should have that data, again, within a time frame of several weeks.
Okay, now moving on to the second aspect, which is treatment, or minimizing the effects once you get infected, unfortunately. I've heard a lot of talk about cytokines. If you can, cytokine storm and so forth. Can you explain to us what that is and how damage is done?
Yeah, so let me just, maybe I'll say a few words to put it into a little bit of context. So obviously there are many questions swirling around COVID-19. And one of the key questions is really, why do some individuals progress to more advanced disease? And that's not just the respiratory insufficiency in the first instance, but there's a litany of other systemic manifestations that a minority, but a distinct minority of patients may progress to. Some of them pretty puzzling, by the way, including the blood clotting, thromboembolism. So, and the general assumption, hypothesis, if you will, out there has been that it's something called a cytokine storm. So really, to answer your question, cytokines is really this broad and pretty loose category of secreted small proteins in the body that are really important in cell signaling and have all kinds of effects. They're produced by a very broad range of cells in the body. And in turn, when secreted and they get in your blood system, they'll go to different organs and they'll have affect a broad range of target cells along the way. In the context of COVID-19, we're particularly interested in this set of cytokines that are called pro-inflammatory. They drive inflammation. And in fact, and they get produced, and all these cytokines get produced as a response to the initial virus infection. And it develops this massive inflammatory response in some individuals, really a hyper-inflammatory state, if you will. And so just as the case of vaccines, there's been this flood of hot prospects of anti-inflammatory agents that making it to the headlines. Some of them have already come and gone. This is a pretty fast cycle. And in order to really parse that and come up with what should be the best, you have to have a clear and distinct point of view on how COVID-19 disease evolves, something that you call pathogenesis. How does the disease evolve in people? And so, and I say that, and now I'm speaking wearing the hat of I'm a former chair of a department of pathology back at the University of Pennsylvania, and I have a point of view about pathogenesis. And often what you find is that, especially when things are happening in a hurry like they are now for COVID-19, some of the clinical trials are just sort of started a little bit willy-nilly and not always well conceived and linked to pathogenesis. And let me just give you an example. If you've got an antiviral, which is meant to stop replication of the virus, then you can't sort of reject its efficacy if you're delivering it very late in the disease when you're already in a cytokine storm and inflammation. It may still have a role there depending on the particular virus and person, but you've got to sort of target and evaluate that. And in a lot of instances, what you see is people have rushed things in there, sort of the easy path. So you'll go at patients who have advanced disease, and then you end up rejecting a candidate of something that could have been effective in an earlier stage of the disease, or vice versa. So timing, where things happen in the body and when they happen and how disease evolves becomes a really important consideration.
Just before we get further on, because we all know what inflammation is, but I maybe not understand it. Why is inflammation bad? If we're producing these cytokines, shouldn't it be good for our body?
Yeah, so pretty much everything is good to some extent, and then too much of a good thing becomes bad, right? So you want to be able to fight the virus, and so vaccines are giving you an immune response, but sometimes that immune response becomes way too robust and then starts creating all kinds of other physiologic types effects. Also, some elements of the immune response are really not helpful for attacking the virus and actually drive pathogenesis. And we generally sort of loosely use the word inflammation as generally a bad thing. You want to develop good immunity and good immune responses, but when it's too much or of the wrong kind, then we sort of label it as inflammation.
Dr. Tykocinski, we've heard that there is some cool new research that you are doing in your lab with your team of researchers. Could you elaborate on that for the rest of us?
Sure. So, for full disclosure, let me say, my full-time day job is as provost and dean of the med school, but I do run a small lab, and I also, a number of years ago, started a biotech company in the immunotherapy space. It happens to be developing immunotherapies for cancer, and we're very close to getting things into trials. So that's my sort of the context of my work. But like literally hundreds, if not thousands of scientists, it has everyone asking the question, okay, is anything I'm doing right now relevant to something like COVID-19? And obviously, in some of this stuff, it's not, we'll hopefully and almost certainly be beyond COVID-19 by the time they make it all the way through. Some of them could be repurposed, insights will lead you to repurposing existing drugs, and there's a lot of that out there. But so we've, my lab has been very interested in the whole set of protein pharmaceuticals. About half of pharma's pipeline now are what's called biologics. They're not small molecule drugs, but they're actually proteins. Antibodies, for example, are an example of a protein. Our angle, and we've had a whole series of patents around this, is making fusion proteins. If you have a protein A with function A and a protein B with function B, if you fuse them together and make AB fusions, can you get A plus B function? And we use that, and one of the things we have done with those kind of fusion proteins is to target specific cells of the immune system and rev them up. And that's really the company that I've already started, which is in the immunotherapy of cancer space. But now, thinking about COVID, we've said, can we make ones that go in the other direction? Can we make fusion proteins that, have some of the properties that we've developed and developed intellectual property over the years to ramp them down? And so we've developed some insights of late into thinking of, amidst this parade of immune cells that are in the body, what might the one or two cell types be that are of particular interest and may actually be key behind the cytokine storm, and those that may be producing those inflammatory cytokines, and then having our fusion proteins designed that will target those cells. So more to come in the coming weeks, but and at least in our lab, we're pretty excited about some of the ideas that we have now come up with around that.
Yes, calming the storm, in essence.
Yes, exactly. And let me just say one other thing, because you'd asked earlier about the timing. There is one solution that comes as in the antivirals before even having a vaccine. And there's been a little attention given to it, but not as much as I think it deserves in the media, which is the monoclonal antibodies. There are a couple of companies now that have reported developing neutralizing monoclonal antibodies. Now, just to understand that, when you do a vaccine, the goal of vaccine is to get the body to produce antibodies against the virus. But as an alternative, you could just produce the antibodies outside the body and mass manufacture them, scale up manufacture of those antibodies, and then infuse those antibodies. We do that in part, you may have heard of, using convalescent plasma. You take, the blood plasma of patients that have recovered from COVID-19, and we're putting back into patients. And there's some promising data that's emerging around that. Well, here you would just manufacture it. And we have a whole industry now around producing monoclonal antibodies, single clone antibodies that do different things. And if we get this fall a set of neutralizing monoclonal antibodies that become available, they could be used alone or individually, potentially even for prevention, but certainly if somebody comes down with the disease, that's sort of a next best thing in a way to having the vaccine, and that alone, I think, could make a big difference out there.
Doc, there's a lot of people, including myself and my family, who have a lot of anxiety, and I'm sure speaking for everyone who's listening to this broadcast as well. You being more on the inside than us, and not being a politician, but actually being a scientist, which counts a whole lot more. Are you feeling optimistic regarding whether it's the antibodies you just talked about, vaccine, or treatment, or all of them together?
Yeah, I think overall, I tend to be an optimist, so I confess. But I do believe that it is, especially because on the way, first of all, we have such a flurry of activity out there going on, as I said, towards the vaccines. And some of those candidates really look promising. And I think that sort of the pathway, I think can be accelerated. But even before we get there, I got very excited about the reports of neutralizing, monoclonals. Obviously, there's a lot of work still to be done. We don't know, it's sketchy reports, so, those have to be validated. But with that work, with a number of other developments in terms of how we might treat and temper the body's storm, the cytokine storm, and other things, I think we're going to be in a good position. Now, obviously, the next couple of months remain of concern, of considerable concern. So when I talk about optimism, I don't mean optimism for June, but I do mean optimism for the fall, into late fall. They're obviously the concern that you'll now have influenza back and the overlap of influenza with COVID-19. So I'm not minimizing the issues around that, but I don't think we've ever seen quite the mobilization of brain power around the planet that we've seen. Unfortunately, we shouldn't have ever been in that kind of position, where we have to sort of do this catch up.
So that brings us to the future. So my question is about that. So when we do a ex post analysis, we always ask, how did this happen? And when you think about the whole world, what lessons can we learn from what we are going through actually? And do you think that there's anything that we as a university, country, or the world can do going forward to prevent something like this from happening?
Yeah, well, the answer is yes. So let me share my perspective on it. We've had ample warning, is what it comes down to. If you just look at the last couple of decades, it's not like we haven't seen epidemics and pandemics. To think that was just a matter of 1918's influenza and epidemics was sort of out of the mind is just simply obviously not the case. We had the swine flu pandemic, the resurgence of yellow fever in parts of Africa, Zika virus, Ebola epidemics, and then even some coronaviruses, right? SARS and MERS. As a global community, and that includes as a global scientific community, but certainly the kinds of things that the Arlen Specter Center is interested in terms of policy, the responses have really never been sustained. Haven't been sort of real system-wide solutions. And at best, I'd say it's fits and starts. So largely, we always seem to be in this reactive mode, and at that, pretty slow to react. Except this time around, it seems to be moving a little more quickly. There's a sort of an interesting story, if I can just digress, because that involves Jefferson in Philadelphia. There's one of our medical school alumni from the class of 1855, Carlos Finlay. He was studying yellow fever, and that turns out yellow fever is a virus, and it turned out to be a major pathogenicity. We don't speak about it much, but in the 1800s into the early 1900s, major issue. There's a recent book that came out on yellow fever in New Orleans by Ernie Willoughby. And Finlay in 1881 discovered that mosquitoes carried it. And yet, everyone sort of sidelined him over this. He was sort of marginalized and kept poking holes in it. And so there was a 20-year period before anybody would even accept that hypothesis, even though by then we also already knew that there were other mosquito-borne diseases. And then it wasn't until 1927 that the virus itself was this, and that we had a vaccine by 1930. Now, so we sort of have this history of alarm bells that we don't believe, and then we don't act on them, and that's in the centuries past, and it certainly continues on till now. It's not to say that foundations like the Gates Foundation and other individuals haven't sort of pushed for that and have been trying to create systems that expedite this kind of thing, but it's really been confined to the few and limited in scope. But to specifically, in brief, to answer your question, there's a call to action here. I mean, I think there's a real call to action, and that call to action says we have to create system approaches that allow us to, first of all, when we have first glimmers of an infectious pathogen out there, anywhere in the world, that take us from streamline the process from pathogen to a vaccine and targeted diagnostics and therapeutics. We have the tools in the toolbox. We just need to assemble them and systematize them and coordinate globally in a way that, which just, in my view at least, we simply haven't done and effectively. And then also the part of that toolbox is, how do you scale up production, right? If you can't do things sequentially. If you wait till, let's say, you validate a vaccine to first start to scale up production of that vaccine, well, that's too late, right? So, and you see that happening now, and there are investments being made at various levels to sort of move forward in the scale up even before we have confirmation that some of these things work, and that's a good thing. And then also, we should be creating entire armamentarium of molecular candidates like anti-inflammatories that we do. And by the way, the idea of cytokine storm isn't new. We need to ask ourselves, why wasn't there more funding? Why wasn't, with all the research, and I'm not saying there wasn't a reasonable amount, but these are solvable problems if you put enough resources into it, and at a time when we're investing trillions now, we see the consequence of not doing that. In the trillions, for a couple of billion, some of these things could be solved instead of having scientists sort of scrounging for the last dollars in their NIH applications. But that's my bully pulpit and that's my agenda. So I hope you excuse me for that.
No, that's perfectly fine.
Doctor, just wrapping things up this one very quickly. I've heard a couple of things I'd like for you to straighten out on social media and stuff. This is just the flu. Can you address that comment that's been made by many?
Yeah, and look, I'm not a viral immunologist or a virologist, so I really want to, have that proviso in there once again. But clearly, from what I see and know and read, I think it's pretty clear to all of us that this is, has a higher R naught. This transmits more actively. There's still a lot of questions in terms of transmissibility from children to adults and all these other kinds of questions. But to me, without a doubt, this is a more transmissible virus. It's also, scarier. Now, some of the earlier, fears, may have, been, turn out to be not as bad, but it's clearly not just a flu. and there's something worse about this one. And you don't want to get that. You generally don't want to get that. The good news is that, young people are safe, and as a parent and as a grandparent, for me, then there's a lot of relief in knowing that. And there's these interesting, tantalizing clues in terms of the comorbidities that make it worse. So, it's more than the flu, but it's probably not as bad as the absolute worst case scenarios had us believing. And there's still a lot of questions to be answered.
Oh, that makes me feel so much better. This one last question: how do we stay safe? And then I'll turn it over to Raju if he has any.
Yeah, I would. Well, I can only tell you. I think we're still in a wear the mask phase, and in a social distancing phase, and the situation will evolve considerably. Clearly, over the in the coming few months, over the summer, we'll continue to learn a lot more. We learn a lot more, frankly, each day about this. But I think until we fully know that, and certainly for individuals that have comorbidities, higher risks, people in older age groups, we want to be really careful. And so, we're planning to reopen our campus in the fall. We went public with that, one of the first actually in the Philadelphia area. But that said, it's with the idea of not business as usual. For those on campus, social distancing, some of our large group formats will be done still online, even if they're sitting there on campus, and we're not alone in that. So, people should stay safe, wash your hands, 20 seconds at least, they say, and we'll get through this.
Dr. Tykocinski, I don't have any further questions. I think with the time that we have sort of hit a lot of the good points here. And I would like to thank you for explaining things in such a lucid, simple manner, and so that the general audience can understand what exactly, for example, is a vaccine. And especially our research that we are doing at Jefferson, including yours. We're quite excited, at least in that sense, for the future, immediate future. But I believe this has been a very informative and very helpful session for everybody tuning in, and also for us to sort of highlight and showcase our research to the rest of the world. I'll turn it to Evan unless you have a comment to that. Go ahead.
No, I just want to thank you for the opportunity, and I think it's just incredible. This kind of format is incredibly important. To have an opportunity to talk about things and really reflect on what's out there, and sort of navigate through the information.
Thank you very much, Dr. Tykocinski, and thanks for being our first host. And hopefully, we'll be talking about much lighter events in further broadcasts. Thank you again.
And thanks to our audience too.
And congratulations on this format. Thank you.
Thank you, Doctor.